Should I start? Okay. Thanks, everyone. I'm here to represent Bratislav Misic. He's the leader of the Network Neuroscience Lab. He did his PhD with Randy McIntosh in Toronto, and then his postdoc with Olaf Sporns in Indiana. And he leads his lab in MNI in McGill. So, well, you'll see his research. I think that talks enough. Thank you for coming.
Thank you, Eduardo. Thank you to the organizers, all of you, for having me, and thank you all of you for coming. And I just want to say we've really enjoyed our time here so far, and it's been a wonderful visit with a lot of super cool research that you guys are doing. So congratulations on that. So I'll tell you a little bit about our work on the structure-function relationship in brain networks. Structure-function relationships are a fundamental question in any area of natural sciences and engineering. Just this idea of how does the architecture of the system that you've made give rise to the different things that that system can do, the different functions. So what I'll show you is not necessarily one single all-encompassing theory that we've doggedly pursued for years and years, but rather because the question itself is so old, we've over the past 4 or 5 years have started to question a lot of the assumptions that we've been making about the nature of these relationships and deconstructing our kind of muscle memory for how to attack these problems. And I'll show you a few different ways in which we've done that. So our jumping off point is, our jumping off point is that my Google Drive is out of memory, but that's going to be, yes, is this. So we know that the brain is despite the fact that we study the brain from a very macroscopic perspective, we know that it's a multi-scale integrated system starting from the gradients of gene expression and that we have across different regions that provide the code and the recipe for how different components are going to be arranged with respect to one another. So where different neurotransmitter receptors are going to be placed, how, and as a result, how synapses will form, how neural elements will exchange signals in the form of electrical impulses, how different cell types are going to be arranged, what types of functions they will specialize for, and the types of neural circuits that they will form. And when we get to this kind of more macroscopic end of the scale, what we're looking at is brain structure, usually for us in the form of diffusion-weighted MRI, and brain function, again, usually for us in the form of functional MRI and the bold response. So we take time series frequently and compute functional connectivity. So I'll show you kind of 3 themes in this domain. One that questions whether we should be looking at structure-function relationships at a global level or whether we should be focusing more on regional heterogeneity and what we would call local structure-function relationships. One where we will try to come up with different ways to annotate our connectomes, our brain graphs, and incorporate additional biological metadata that we have on those graphs. And then finally, we're a set of studies where we've reconceptualized this or questioned whether we should really be looking at functional connectivity per se, but whether we should be looking more specifically at computational functions. So various things such as vision motor integration, and so on. And there I'll be talking about this idea of neuromorphic networks. So these would be bioinstantiated artificial networks that have been constrained by connectome diagrams. So just so that we're all on the same page, I suspect that most people know this, but when we talk about connectivity, we're really talking about 2 different modalities. With structural connectivity, what we're doing is we're parcellating the brain into a set of gray matter nodes, and then using some form of diffusion-weighted MRI or track tracing to reconstruct the white matter fibers that interconnect different parts of the brain to one another. This gives us a set of these matrices that we call adjacency matrices, and the elements in these matrices tell us the probability that any 2 elements are connected with one another. And at the same time, we can take the same nodes and we can record neural activity, either electrophysiological physiological or hemodynamic neural activity, and look at how, for different pairs of brain areas, how coherent their time courses are. The idea being is that if two areas have similar time courses, then presumably they're engaging in some common function. Maybe they're even exchanging information in some way. So if you compute all pairwise associations between these time courses, you get your functional connectivity matrices. And the main thing to keep in mind, obviously, is with structural connectivity, we're talking about the physical infrastructure, something that's very stable, relatively sparse. And with functional connectivity, we're talking about more of a statistical concept, one that's dynamic across time and can take on multiple configurations. So the nature of the problem is this, and that is that you saw very nicely in the previous talk, and that is that with functional connectivity, you can obviously have connections between all possible pairs of brain areas, but only a small subset of those are actually going to be subtended by an anatomical connection. We know that those pairs of brain areas that share an anatomical connection are going to display stronger functional connectivity. That's what you see there. And we know that if we take pairs of brain areas that have both a structural connection and a functional connection, we correlate those, we get a positive association, meaning that areas that have greater structural connectivity, connections that have greater structural connectivity also tend to be associated with greater functional connectivity. We expect that. But the main thing is that there is a lot of variance here in functional connectivity that is not being explained by the underlying direct structural connectivity. And as Marta said in the previous talk, that's because of polysynaptic communication. You could have lots of different routes by which signals could propagate between two areas, and they don't necessarily have to go directly. So that picture becomes murkier. This is just to show this idea as well, how it kind of translates to the network level, where if you compare the optimal partition into communities or modules of the structural network and you superimpose that on the functional network, you see that it's actually not a very well-fitting solution. And correspondingly, if you look at the partition into functional networks, so-called resting state networks, you see that that's not a good solution for structural networks. So this phenomenon exists at the network level as well. The way that we've thought about the problem is that with structural connectivity, you have the physical infrastructure that supports the propagation of electrical impulses between pairs of brain areas, and the superposition of many of these elementary signaling events ultimately manifests as patterns of interregional covariance that we call functional connectivity. So these two domains are empirically accessible to us. We can measure them. But the nature of this communication process is something that we and others have tried very hard to understand and to model. And just to give you an idea, just one study of how this has typically been done, and I'm showing this because I'm very familiar with it, is this. We've proposed a particular model. This is a percolation model, so it's a form of diffusion, where we said, well, in a network, if you have a perturbation, this could be, for instance, the transduction of a sensory stimulus, that perturbation is going to affect a lot of the connected neighbors probabilistically, and is going to eventually spread through the network and develop into a signaling cascades that envelops some nodes and not others. So this is a really simple model, but a powerful one that can allow you to look at many different, potentially biologically meaningful situations, such as where you have this transduction of a single stimulus, where you have two stimuli that come in but carry the same kind of content, so say binaural stimulation or something like this, or you could have competitive spreading, where you have two stimuli with different content and then spread through the network and compete for influence. So for instance, you're hearing two different streams of auditory input or something like this. And then the typical way in which these models are used is, one, you kind of analyze what they tell you about the brain. So you can look at, in this particular model, what classes of connections are most likely to be utilized. They tend to be a lot of these so-called connections between the so-called rich club nodes. You can look at which nodes are going to be the meeting points for many different cascades. So presumably the areas that are most highly predisposed to integrate information from multiple sources. So for instance, here what you see is that this map is particularly enriched in the default mode network. So it tells you that a lot of the integrative properties or character of some of these transmodal networks just naturally arise from the way that they're embedded in the global anatomy. And then the main thing that I wanted to show you is that you can use this model to try to predict functional connectivity. So for instance, these are patterns of functional connectivity that we have in the same subjects that we ran this model on, structural connectivity. And this is the functional connectivity that you predict from the model. So we looked at how frequently areas were likely to be co-activated in these simulations. And the main thing here is that you can get a pretty decent prediction of functional connectivity using models like this. It's not perfect, but the key thing is that actually it's roughly the same as what you get with very complex, very detailed biophysical models that have lots of parameters and entail a lot more assumptions. This is a very simple model that so the assumptions here, I think you can already start to see, and that is that there is a single global model that should be applied to the whole network, and also that all nodes are going to be are going to have kind of the same identity in a way. So let's just going to get into that a little bit and talk about this idea of regional heterogeneity. So the two big assumptions, as I said so far in the literature, I think, are that the nature of this structure-function coupling is global, and that there is always a single optimal model. So I showed you one model that we were interested in, this idea of percolation. Somebody else could come in and say, there have been people who proposed navigation models, very complex biophysical models, statistical models, things like this. If you look at other systems where you have to achieve communication over a resource-limited medium, such as, for instance, telephony, we know that multiple communication protocols can be utilized over the same medium. And so what we thought was, well, maybe what we should start to think about was a setup like this, where we have multiple reasonable models of communication that are simultaneously being played out on our network to give rise to functional connectivity. And so I'll show you a study where we did both of these things. This is kind of a group average structural connectivity network, group average functional connectivity network. And what we're going to try to do is to predict the functional connectivity from three different predictors based on structural connectivity. One is simple Euclidean distance associated with each of the connections. So this will be consistent with navigation. Path length, which is consistent with shortest path routing, and communicability that's consistent with diffusion. So these are kind of the three canonical axes that separate all the different communication models that have so far been proposed. And the other thing that we're going to do is we're not going to do this for the entire network. We're going to say, well, maybe some areas tend to communicate in a particular way, and some areas tend to communicate in a different way. And so what we're going to do is we're going to take a row of our functional connectivity matrix, so the kind of the connectivity profile of one region, and we're going to try to predict it as a function of all of these other structural predictors. So this is a really stupid, simple linear regression, multiple regression, and we can do it for each node. So what you're going to see first, what we looked at first was if we do this for all the brain areas, and we're going to look at this structure function coupling R squared. So how well can we predict function from structure in different areas? The first thing that you see is that there's a hell of a lot of variance in here. So for some areas, we can predict function pretty well, and for some, we do very poorly, even though we're probably overfitting anyway. If you look at where these areas are, this diagram has been drawn in such a way to accentuate those areas that have particularly poor structure-function coupling, so where this fit is very low. And you can see these are some of the most exciting areas in the brain. A lot of what we tend to think of as polysensory association cortex. So you have medial parietal, a lot of kind of lateral parietal, insular cortex, and so on. And we thought that this looked a lot like the unimodal transmodal gradient that Daniel Margulis, who I believe gave a talk here some years ago, had kind of proposed, and he came up with a really simple method to extract this from functional connectivity. So this is that image, and this just separates the brain into unimodal cortices versus transmodal cortices in a continuous way. And so when you correlate the two, you see that there's a really nice negative relationship. In other words, in unimodal cortex, it's very easy to predict function from structure, and in transmodal cortex, it's very hard to predict function from structure, and this seems to be a systematic effect. And this was really cool, and I'll come back to this idea shortly, but we knew of this paper by Randy Buckner and Fenna Kreenan, who talked about this idea that due to the rapid evolutionary expansion of these transmodal cortices, you start to get a sort of a release or an untethering from the underlying strong genomic gradients. As a result, you start to get more complex circuit configurations that presumably do not just support simple kind of sensory motor responses. And that's an idea that I'll come back to in a second, but we thought that was kind of interesting. I just want to say that we can also look at this from a dynamic perspective. So there are a number of ways in fMRI that you can look at dynamic functional connectivity. So looking at how functional connectivity changes frame by frame. If functional connectivity changes over time, then presumably structure-function coupling can change over time too. So we used the exact same method that I just showed you, but looking at dynamic functional connectivity as the dependent variable. And what you're seeing here is how variable the structure-function relationship is. Yes? If I can speak more like this, I apologize. I don't usually do this. Just bar mitzvahs and other things, right? Yeah, so what you're seeing is how dynamic structure-function coupling is for different areas. We're looking at the coefficient of variation. So this is standard deviation scaled by the mean. This is an example of two areas, one I believe in insular cortex and one in parietal cortex. You can see the nature of these fluctuations. But what you're seeing here is if we stratify this map into different partitions of the cortex, exactly how this looks. And now it's not a unimodal transmodal gradient at all. It's something that actually peaks in the middle of this hierarchy. The middle of the hierarchy is typically insular cortex. And indeed, if you divide the brain up according to the Yeo-Kreenan networks, you see that this map is enriched in the ventral attention network. So again, insular cortex. And if you compare it to the von Economo cytoarchitectonic classes, again, what he called insular cortex is highly enriched there. So what we're seeing is this cool effect where insular cortex was just thought to be kind of the midpoint between interoceptive processing and processing of external extrinsic stimuli is the particular area that's fluctuating most in how it mediates this link between structure and function. It is also cytoarchitectonically very diverse, which was interesting. But and part of the reason, at least physically, we think, is if you look at the total length of connections for different parts of the brain, you see that the areas that have the kind of the most, the nicest mix between short and long distance connections that are most intermediate here, they tend to actually have the greatest variability in this structure-function coupling. So there are lots of effects here at play that bring into account geometry, presumably the underlying cytoarchitecture as well. And that's something that we'll pursue in a bit. Okay, so this whole kind of set of studies, and there are others, but these are good examples, kind of led us to start to think about, well, what is actually different about these areas, and could some of these systematic differences in structure-function coupling actually be due to differences in cytoarchitecture or other underlying biological features that we're not looking at with our connectome reconstructions. So we started to look more at annotated networks and trying to attach different biological features to the connectomes that we're making. And this is just to illustrate this. This is how I grew up, sort of seeing the brain. It's a gray network with gray nodes and gray edges. But what we know is that these nodes are not actually gray. They have some color. That color could be any number of features that are very microscopic, such as gene transcription They could be side architecture, they could be neurotransmitter receptors, and a lot of other microstructural features that we can get with garden variety MRI scans. So this kind of opened up a program of research for us where we tried really hard to look at how the local annotations actually interact with network embedding. And so, yeah, oh, this is so I'll show you one thing, and again, a good example of how we've started to deconstruct what we're thinking about when we think about structure and function. Function or functional connectivity is really a function of the time series of the constituent nodes. It's a correlation coefficient between the time courses. So maybe we shouldn't really be looking at functional connectivity at all. Maybe we should be looking at the time series themselves. This is not a new idea, obviously. In electrophysiology, this is very common. In fMRI as well, people have, there's a rich literature of people looking at things like standard deviations, entropies, things like this, intrinsic timescales, and so on. The only thing is about that literature is that people will pick their favorite time series measurement and they'll just do that. Or they'll pick something that somebody else has picked, just so that they kind of have something to write about. So we were very much inspired by this idea of looking at how the local circuitry changes across the brain, but also how does that affect the dynamics in the time series. So what we did was to take data from the Human Connectome project. We took all of the resting state bold, and we used something called highly comparative time series analysis, a technique that Ben Fulcher had developed. And the idea here is that you take a time series, you submit it to this toolbox, and it calculates the 6,000 or 7,000 different time series features known to man that have ever been invented in physics. So anything that has to do with fluctuation analysis, entropy, different power spectral measures, entropy, autocorrelations, so on and so forth. So it gives you a really detailed, really comprehensive dynamic fingerprint for every brain area. We can do this for each of our regions. And then one thing that we did that I'm not going to talk about too much is we looked at how similar regions are in terms of their profiles. But we also looked at just what the dominant low-dimensional components of this representation are. And I think it's very instructive. So not to kind of go into too much detail, but what we found were two dominant components. One that really separated ventromedial and kind of dorsolateral cortex, and one that ran in a very stereotyped kind of unimodal, transmodal kind of fashion. And if you look at the time series features that load most highly on each of these components, you quickly begin to realize that with PC1, you're looking at measures of autocorrelation, and these are just two examples of highly loading features, LAG1 autocorrelation and nonlinear autocorrelation. And I've picked a few choice regions here just to show you how the time series change. So this is kind of a well-known phenomenon that in some of these, especially parietal cortex, you're going to get larger autocorrelations, so you have longer intrinsic time scales. And here in this unimodal transmodal kind of gradient, what we noticed was that we kept getting time series features that had to do with the distribution of the signal amplitudes. And in particular, they had to do with the even moments of the distribution. And as many of you probably know, even moments tell you about how evenly distributed the distribution is around the mean. So what you're seeing here is that this unimodal transmodal cortex, this kurtosis, or the fourth moment in distributional entropy, you see that the signal amplitudes start to get stretched out more and more. In other words, for instance, here in unimodal cortex, you tend to have a greater dynamic range. These time courses can take on a wider variety of values, and in transmodal cortex, they have a lower dynamic range. So two things to keep in mind, or to kind of point out here. One is that, for instance, this this gradient of autocorrelation, which was it is widely discussed in the literature as a unimodal transmodal gradient, is actually not a unimodal transmodal gradient. It's more of like I said, a ventromedial dorsolateral gradient. And this other gradient here, the unimodal transmodal gradient, which is thought to be primarily a gradient of connectivity, can actually be recovered using a much more fundamental set of time series features. Now, the cool thing is that these two signatures of dynamics actually have very strong underlying microarchitectural correlates. These are what we had at the time. Since then, we've expanded, and I'll show you some of that tomorrow. But basically, the first component of gene expression really nicely predicts this dominant gradient of autocorrelation. And this second gradient of dynamic range is nicely predicted by the other measures that we had, so T1, T2, cortical thickness, and so on. So it just shows you that as the underlying local circuit properties change across the cortex, you're going to start to get different dynamics, and that could easily be driving a lot of what we see with functional connectivity. Okay, now, right. Then we did something really kind of insane where we we just said, well, we were literally looking at a drawing like this, and we said, well, you know, why don't we just go all the way to the left, look at gene transcription, and why don't we go all the way to the right and look at function, and whether we can find genomic signatures of different psychological processes. So that's what we did here. We're looking at gene expression from the Allen Institute Human Brain Atlas. This is a comprehensive atlas of microarray transcription for 6 postmortem brains. And we're looking at an atlas of functional activation across many tasks, the so-called neurosynth atlas. So this is a meta-analytic atlas that tells you the probability that different brain areas are activated with respect to certain cognitive terms. So what we did here was to take our gene expression atlas, so we have nodes by genes and their expression level, and we took this neurosynth atlas that had nodes by different cognitive terms. We then correlated them and subjected them to singular value decomposition. For those that many of you will recognize this as a partially squares analysis. And the analysis is going to give us some set of gene weights, so weighing different genes, and some set of cognitive processes that optimally covary with one another. So we're going to look for cognitive phenotypes and their underlying genomic signatures. And we're going to ask, first of all, can we identify such pairings? And second of all, what do they look like? So the main thing that we saw was, if you first look at the terms, so always easier to look at the cognitive terms, is you get essentially a gradient that takes you from higher kind of cognition and attention and things like this, all the way to emotion and affect, so kind of a cognitive affective gradient. And the genes that support this, this is kind of like, these are word clouds that show you what functions they're engaged in, but it's what we did that I think is probably the most instructive is we did cell type deconvolution. So we looked at this genetic signature at the different ends of this gradient, and we said, what types of cells are most likely to be expressed here based on the gene weights that we're getting. And what you're seeing is that for the kind of the more quote unquote cognitive attention gradient, you're getting an overexpression of neurons, inhibitory neurons, excitatory neurons, and so on. For affect, you're getting an overexpression of support cells, endothelial cells, oligodendrocytes, microglia, and so on. So it's a nice example, I think, of how you can completely circumvent structure and function, or structure and functional connectivity as we do it, and look for the most basic, the most microscopic signatures of broader cognitive processes. We know that this, we also show in the paper that this gene term axis strengthens in development, and we replicate it in BrainsPen and BrainMap. Okay, so now that's all fine and good, gene expression, cell types, and so on. I think, like, personally, my bias, if I was to look at the biological annotation that I think would be most meaningful for understanding structure-function relationships, is the distribution of neurotransmitter receptors. So the idea that how this could play out is you could have ascending projections, say, from the brainstem, which you can now segment, measure, even trace out. And based on which nuclei are sort of engaged and which projections are being engaged, you could induce totally different types of signal exchange in the same underlying network. You could have a situation like this where signal exchange among within kind of modules of nodes that share similar functions is promoted, so it's kind of more segregative. Or you could have a situation like this where exchange among brain areas that belong to different systems is promoted, and you can have greater integration. So the distribution of neurotransmitter receptors is going to condition what types of functional connectivity configurations can arise from the underlying anatomy. So to address this question, what we did was to basically start cold calling all the different PET researchers that we knew locally and globally to ask if they would be willing to share with us the data that they had on different neurotransmitter receptors from PET imaging, positron emission tomography. After kind of a lengthy process, we accumulated a data set that had information about all of these different neurotransmitter receptors and transporters. So it's over 1,200 different maps that encompass 19 receptors and 9 different neurotransmitter systems. And then we asked, how does this map onto structure and function? What we know is that, well, first of all, the main thing that we were doing is taking pairs of areas, looking at their receptor profile, and then correlating these receptor profiles to get an idea of how similar the chemoarchitectural profile of 2 areas is, and whether they'd be whether they would be influenced in the same way by ascending projections, for instance. This gives you a region by region receptor profile similarity matrix. We know that these values in this matrix, that there's an exponential drop-off in receptor similarity with Euclidean distance, so areas that are further apart will tend to have increasingly dissimilar receptor profiles. This is just kind of a sanity check. We then reference this against structural and functional connectivity that we had from the Human Connectome project. You see right away, actually, that receptor profile similarity within, oh no, you should not be looking at that. So one thing with structural connectivity is that areas that actually display a structural connection, they will tend to have significantly greater receptor profile similarity. And areas that are part of the same resting state network, that are part of the same cognitive system, will also tend to have greater receptor profile similarity. So areas that are connected and participate in similar functions tend to have more similar chemoarchitectural profiles. And this metric is also just plain correlated with structural and functional connectivity. But the main thing that we wanted to do was to ask whether incorporating this additional metadata would actually help us to predict function from structure. So now we're going all the way back to what I was showing you before, these maps of regional structure-function coupling. This is just using a slightly different color map, but again, you can see that structure-function coupling is greater in unimodal cortex than transmodal cortex. And I'm going to compare this to a set of structure-function coupling maps where I'm doing the same thing that I did before, but now I'm additionally using receptor profile similarity to see how well I can predict functional connectivity. And what you can see here is that generally adding in this receptor profile similarity increases my prediction of function from structure for pretty much all areas, and the ones that are shown in orange are statistically significant. This is the identity line right there. These are the areas that, in case you're interested, these are the areas that benefit most from incorporating this information. As I said in Querétaro, I don't know whether this is because I'm simply covering the gaps either in my structural connectivity reconstruction or in my functional connectivity, or if I'm uncovering something that's really different for the neurotransmission that we see in these areas. But nevertheless, adding this data improves prediction. Okay, so that's pretty much all I had on annotated networks. And then just for the last part, I wanted to talk about a bit of a departure, actually, from all of this, and to tell you about how we've really thought about not rethinking structure, but rather rethinking function. When we talk about function, or at least when I talk about function, and when we've written about this in reviews and so on, we've used function as shorthand for functional coactivation, really, for functional connectivity. But that's not necessarily what we want to understand in this wider program of research. Rather, what we want to understand is how the underlying architecture actually gives rise to different types of computations. And so that's where we started to look at this technique that exists, I guess you could call it AI, and it's called reservoir computing. And the way that it works is you start out with an artificial neural network. Typically in that literature, they're completely random recurrent neural networks. Each of these nodes is a dynamical system, so it has some activation function. And the way that it works is you have a set of input nodes where you transmit or beam in some stimulus, and then you read information out from a set of output nodes with usually a linear classifier. And the job of the artificial network is to try to learn some desired function of the input signal. So for instance, you have an input signal here. The dynamics are activated, and then based on the outputs of the linear unit, you might want to, you have some output. And what we did in our case was to reconstruct a time-delayed version of the input signal. But actually, I'll talk about that in a second. But this is the broad idea. In our work, what we did was to use a set of the hyperbolic tangent activation functions. So it really boils down to discrete time recurrent neural network of spiking neurons. And the main thing that we're going to do that's different from what's typically done in reservoir computing is rather than using completely random networks, we're actually going to input connectomes, and we're going to take biologically realistic connectivity patterns, and we're going to teach them to do different cognitive tasks and ask how well does the architecture of the brain actually support different types of computations. So this is how it works. We have a network, a brain network. We input a signal. In our case, this is a uniformly distributed time series. And this time series is fed into a set of input nodes. We've done this many different ways, mainly at the request of the reviewers. And we've done this with inputting nodes into various subcortical, inputting information into various subcortical regions, into primary sensory cortex, et cetera, et cetera. And it generally works the same. Because of the architecture and the dynamics of the nodes, you have some set of internal states, and then a linear classifier is trained, in our case, to represent a time-delayed version of this signal. So what we're teaching the network to do is a working memory task. If I give you a set of stimuli, can you remember the stimuli that came 1 step, 2 steps, 3 steps, 4 steps after? So for every time we do this, we're going to have some prediction accuracy. And the main parameter for us that we're going to tune is what you'll sometimes see as an alpha in some of my diagrams. But we're scaling the spectral radius of the network. What that means is we're scaling the adjacency matrix by the largest eigenvalue. And that has this nice property where we can actually tune the dynamics. So we're going to have for each of our kind of parameter values, we're going to have a memory capacity, a performance, and we're going to have a curve that looks something like this. And this tuning parameter allows us to switch between completely stable stereotype dynamics and chaotic dynamics. And when we have a spectral radius of 1, we're at that hypothetical sweet spot where we have criticality and critical dynamics, and the system is presumably able to take on a wider range of values. So the main question that we're going to ask is, how do the different resting state networks actually support memory capacity. So, resting state networks are widely studied. They're thought to be the building blocks of higher cognition, but really they're a solution that comes out of an independent component analysis or a community detection analysis. So what we're going to do is we're going to pick the different resting state networks as readout modules, and we're going to ask a number of questions about how well the network does this particular task. So what you're seeing here is memory capacity. You're seeing this parameter that tunes the dynamics. This is how well the network performs. This is not surprising that right at the edge of criticality, we have maximum performance. But the question that we wanted to ask, first of all, is does the architecture of the brain actually help you to do better in this memory capacity task? So to address this question, we used a null model where we rewire the brain. And the second question that we're going to ask is, does this breakup of regions into these putative resting state networks that everyone studies, such as the default mode, does that actually confer any computational benefits? And to do that, we're going to use a set of permuted intrinsic network labels, or so-called spin tests. I'll talk more about this tomorrow, but the idea here is to address the first question, we're using degree-preserving rewiring. So we're taking pairs of edges, and we're randomly rewiring so that we create new networks that have the same number of nodes, same number of edges, and same degree sequence, but the topology has been destroyed. So this will allow us to ask whether the architecture of the network is special. With spin tests, what we're going to do is we're going to take the labels of how different nodes are assigned to different intrinsic networks. We're going to project them to a sphere. We're going to randomly rotate the sphere and bring it to the surface of the brain. This will allow us to ask whether the identity or the affiliation of a brain region to a particular resting state network actually is important for how this is done. So this is what you get when you run these null models. You see that most of the time, the performance for the real brain is actually not better than a lot of these null networks, except for this part of parameter space right here at criticality, where numerous studies have suggested the brain actually operates. This is what these memory capacities actually look like at alpha equals 1. So it tells us that the architecture of the brain, first of all, actually does give you an extra boost in this dynamical regime to actually do this task. And second, the arrangement of different brain regions into these intrinsic networks actually serves a purpose. Again, it allows you to segregate information in a particular way that promotes greater memory capacity. The last thing that I want to show from this is that one thing that we were doing when we were doing these rewired nulls, this is just the same result that I just showed you. With rewired nulls, you're actually giving them an unfair advantage because the connection length distribution of the real brain looks something like this. You tend to have a prevalence of short-range connections. When you randomly rewire edges, you can place them, you typically tend to place them on average between areas that are actually further apart. You tend to have a much greater set of edge lengths. So these networks have this unfair advantage where they tend to have much longer connections, and presumably that entails greater metabolic and material cost. So if you take these performance values and actually normalize them by the wiring cost of the network, you see that real brain networks outperform rewired nulls across the whole parameter range. What this means is that not only can we use brain networks to potentially make better artificial networks, or use neuromorphic networks to make better software, we can actually use the principles that we know about connectivity in real brains to make better hardware, to make neuromorphic chips, because there actually where you place connections does also entail a cost. And then if you're interested in running this, we've recently developed a toolbox that is called Con2Res, so Connectomes 2 Reservoir Computing. It's a toolbox that will allow you to do everything that I just showed you, but it will, and it's all already up and running actually. It involves multiple dynamical systems, so you can also use rectified linear units, leaky, integrated fire neurons, things like this. You can also have membristors, if you like. It involves multiple tasks, so we've actually translated something like 20 different tasks from language comprehension to spatial navigation and so on. So you can do the same thing. And what we're doing right now is we're doing this to try to understand how different computational functions evolve. We're using this wonderful data set from Yaniv Asaf at Tel Aviv University, who's done ex vivo diffusion MRI in 120 different mammalian species. And so we're testing all kinds of things like lemurs and kangaroos and elephants, and we're training them to do different cognitive tasks. Okay, so just to summarize then, I hope I've convinced you that network structure shapes dynamics and promotes behaviorally relevant communication patterns. That there's this incredible regional heterogeneity that we've typically not paid attention to, but that is involved in a perpetual interplay between with the global dynamics and the global connectivity. And that we've gone through this process of rethinking what we mean by structure, what we mean by function. We focused more on just making our models more regionally heterogeneous, on annotating them with biologically meaningful information, and we focused increasingly on not just on co-activation, but on learning and computational capacity in these connectomes. So this is our group, all the students and postdocs who did all this work, and thank you very much for your attention. I'm happy to take any questions.
Okay. Any questions, anyone, about these methods? No? Too clear? Luis? I think you need your mic. I can bring this one.
No tengan miedo de hacer preguntas, todos. Just warming everybody up to ask questions. This last part was very intriguing, super fun. I think I sort of got the gist of it when you showed about the toolbox, how each node it's a dynamic system. But I still don't get how the signal can change at each node, how it can be filtered, like trying to mimic more of what the brain does. For example, if you are registering primary sensory cortices, you can essentially reconstruct the sensory stimulus just by registering those neurons in a monkey or in a rat. If you go outside of primary sensory cortex, you can't do that anymore. That means there was some filtering of the data going on at primary cortices. So how much does these dynamic systems at each node change the input signal to propagate to the rest of the network?
Yeah, so it's all being driven by the connectivity, but also by the underlying dynamics. We went with a very simple, initially when we started to do this, because we were dealing with very large nodes, we used neural mass models. So these are very complex models that are constructed usually as nonlinear differential equations. What we found eventually was that we could make a lot of the same inferences using simpler models, and that's why we went with these hyperbolic tangent functions that are more typically used. So that's how it works. It just depends on the connectivity and the dynamics. Whether the filtering is veridical, whether it's actually biologically realistic, we don't really know. We actually can record the time series as the simulations progress, and we can actually see what happens to a representation as you go up the visual processing hierarchy. We've just never done that. It's a great question, and then you can get even fancier. You can there's a variety of tasks that give you even more resolution to do things like this. There's a lot of vision tasks that actually have 2D images where the network has to do something with the pixels and has to actually pay attention to the to the 2D kind of spatial autocorrelation in the image, things like this. Does that answer your question?
Just a follow-up. So in these neuromorphic networks, you are allowing the network to use its entirety of nodes and edges to compute a task, right? Or to have an end product. But in the brain, you can have a subnetwork to perform a task. For example, visual stimuli will not need motor, probably, motor nodes. Do you find the same here, that you find subnetworks, or is everything executed all at once?
Absolutely fantastic question, and clearly I wasn't clear enough. So what we did was to read out specifically from either the default mode or from the visual network, and that's what we were testing. What's also in the paper is we can compute kind of the encoding and decoding capacity of different networks. So for instance, we find that the so-called limbic network, which is centered right around the entorhinal cortex, at least in that partition, actually has the greatest encoding information capacity, roughly in line with what we think that that does. So we did look at how different components are interacting, what they're good at doing, what they're bad at doing, but there's still a lot more to be done. Yeah, I didn't show it in the interest of time, but yeah.
Any other questions from everyone before I ask mine? Okay, Alonso.
Yeah. We comment, first, thank you very much for the talk, very interesting. So you comment a little about this yesterday. So we were talking yesterday about the problems that we face when we are doing tractography about the false positives. You say that maybe the error is spread all around the network and maybe there's no problem. So because I'm not familiar with the functional data, my question is how the errors are distributed there, and if you think that, because you are looking for correlations in both data sets, so you think that the errors behave similarly in functional MRI, and if you know about the efforts of the groups trying to reduce the effect of the noise in functional. Yes. Thank
you. That's a great question. I mean, the truth is that we have no ground truth for functional connectivity. So, and I think it's the efforts usually to people who've done who've used structural connectivity to try to validate functional connectivity. I think that's not exactly the right way because they're different things. So we don't necessarily know, but what I can tell you is that we typically pre-process a billion different ways. So almost all the work that I showed you has global signal, no global signal, different parcellations, different motion correction methods, things like this. The other thing that I was going to say is that, right, how the errors are distributed. Again, I didn't show it because it's like a little vignette, but when we did this kind of comprehensive time series phenotyping, what you see is that this limbic network that is really just a set of nodes that have extremely noisy time series, and it's actually using this method, it's actually very easy to pull out that network. So I think that using potentially just looking at what the time series features are could be a way forward, but in the absence of any ground truth that I know of, I think many methods will tend to either try to focus on test retest or things like this, but we just don't know. I don't know how other people feel about that. But
anyone else? Yes.
Great talk, amazing results. And I was just curious if you ever had a look at the redundancy of pathways, because it seems that there could be the same regions doing the same thing, or similar pathways, but there is also segregation of function. So I wonder if you could, and if there is a way of looking into that.
Yeah, a fantastic question. That's something that's actually occupied the majority of our thinking. When I talked about the different kind of graph theoretic metrics that we use, I showed you something like shortest path length. So path length is the most basic thing that you can compute for if you're talking about interregional communication. It's literally the shortest set of contiguous edges between two areas. We've often thought about this issue of redundancy, or a question of redundancy, from the perspective of, do I actually want to be simulating any kind of communication process that leaves a huge part of the brain doing nothing? So for instance, most path-based reconstructions of the brain, and then all the associated statistics that you see in graph theory, like betweenness, centrality, that people used to look at hubs, closeness, and so on, they will typically capture or involve only about 20 to 40% of the available connections. And so if you compute measures like this, you're making this implicit assumption that the rest of the brain is just kind of vestigial and doing nothing. So I don't know, whether there are actual redundant paths or not. I can definitely identify them using this technique, but the way that we've thought about it is we don't want to be dealing with anything that makes what I think is a fairly unrealistic assumption that only X% of the brain is ever being used. So I don't know how you think about that problem.
Okay, any more questions? No. Do we have time for another one or not? It's 12:30 already. I'll just ask quickly. I have many questions, and it was a great talk. I guess the main one I have is, well, let's just ask this one. How about, can you do this with MEG, for example? I mean, you have nodes, you have MEG data. Have you planning to do this?
Yeah, the paper's written. Yeah, we've done it. Yeah, you can do it, and you get really cool results. Actually, yeah. The criticality? No, we haven't done that. We've done the structure function mapping. Yeah, so we have not done that with MEG, but you could. It'd be very straightforward.
Well, thank you, and thanks everyone.
Thank you very much, Bratislav. Un anuncio importante, importante para el dueño. Hay una camioneta Ford en Valenciana